The Alignment Viewer component in version 3.0 of the CATUI is one of the most advanced RNA sequence alignment viewers available. Currently, the Alignment Viewer is capable of scaling to support the visualization of RNA sequence alignments with more than 100,000 sequences on a laptop computer, a feature no other sequence alignment viewer or editor currently available can claim. The enormous scaling capability of the Alignment Viewer is a result of its viewport-based design. In virtually all sequence alignment visualization scenarios, the on-screen view is generally much smaller than the total sequence alignment. Using data virtualization techniques, it is possible to only load into memory the portion of the alignment currently on screen and still provide a robust visualization experience. Let us now take a closer look at an open Alignment Viewer window on an ARCAD data source. The identification of the data source associated with the Alignment Viewer appears in the tab when docked in the Data Source tab view pane or in the title bar when floating independently. By default, a row label column appears to the left of the sequence alignment data. The row label column can be resized and/or repositioned. The Alignment Viewer status bar indicates the total number of rows and columns in the associated sequence alignment data source. The sequence alignment data source associated with this Alignment Viewer window has 138 rows or sequences and 360 columns. The Alignment Viewer status bar also indicates the location of the current selection within the sequence alignment data source. In this example, I've selected column 145 in row 9. The font typefaces and sizes for the sequence alignment data and the row label column can be set independently. Fonts for the sequence alignment data are limited to fixed width. I can designate any sequence in the associated sequence alignment data source as a reference sequence. Any reference sequence is pinned to the top of the view. Equivalence to the reference sequence in any column is indicated by shading. Multiple reference sequences can be designated. Reordering the reference sequences alters the equivalence shading. The reference sequence can be used for navigation purposes. Control plus F3 moves the selection to the column of the next reference nucleotide. Skipping any intervening gaps in the reference sequence. Control plus F2 moves the selection to the previous reference nucleotide. Also skipping any intervening gaps in the reference sequence. For RNA sequence alignment data sources that support it, the Alignment Viewer can group the rows by evolutionary relationships. Here, our RNA sequence alignment data source is an ARCAD data source, which supports evolutionary relationships grouping, so let's enable it. With evolutionary relationships grouping enabled, we can now navigate the Alignment Viewer across the sequence alignment data source in the vertical direction using the taxonomy browser. First, let's open the taxonomy browser window. By default, the taxonomy browser opens as a floating window. However, we can dock it to the CATUI main application window. The taxonomy browser initially expands 3 levels from the root, but this setting is easily altered. Let's expand it to 5 levels. We can click on a node in the taxonomy browser and the alignment viewer is automatically scrolled to that node. We can set the taxonomy browser to select all rows in the alignment view that belong to a specific node. The taxonomy browser window displays information about the RNA sequence alignment data source the alignment viewer is associated with. For example, node Euryarchaeota has 6 sequences, but node Halobacteria has 0 sequences in the sequence alignment data source. The distribution of all sequences in the sequence alignment data source across the set of evolutionary relationships is also displayed in the taxonomy browser window. At the root, the total number of sequences, 138, matches the total number of rows in the status bar. Moving to node Euryarchaeota, only 117 of the total 138 sequences are grouped within that node or lower. In the alignment viewer, the bird's eye viewer provides the capability to navigate in 2 dimensions using a lower resolution bird's eye view of the RNA sequence alignment data source. Let's open the bird's eye viewer. Similar to the taxonomy browser, the bird's eye viewer appears in a separate window, which can be docked to the main window. The bird's eye viewer provides a low-resolution, high-fidelity map of the sequence alignment data source. Instead of rendering individual nucleotides as text characters, a bitmap image is created, appropriately scaled to match the total number of columns and rows in the sequence alignment data source. 1 pixel in the bitmap corresponds to 1 cell in the sequence alignment data source. Pixels in the bitmap are shaded according to the nucleotide that appears in the corresponding cell of the sequence alignment data source. The current sequence alignment viewer viewport is represented as a shaded box on the bird's eye view. Dragging this box moves the sequence alignment viewport accordingly. Any alignment viewer window can be split into 2 panes. This is called the split screen view mode. The 2 panes are capable of scrolling independently in the horizontal direction, but their vertical scrolling is synchronized. By dragging the splitter in between the panes, the relative size of the 2 panes can be adjusted. In the future, this feature will be utilized to overlay structural relationships between distant parts of an RNA sequence. The CATUI application has an integrated help document that explains in detail how to use the CATUI. The integrated help can be accessed from the help menu or by hitting the F1 key. This concludes our brief introduction to the CATUI.